Hello, everyone, and I want to thank you for joining us wherever you are. Good morning, good afternoon, or good evening, and joining Defeat Duchenne Canada for another in our annual series of webinars with industry updates. And today, we're delighted to be able to present the latest on research and clinical trial updates on Duchenne from Dyne Therapeutics. We'll hear the latest on Dyne 251, the exon 51 skipping therapy, and we'll also hear details about the DELIVER clinical trial. My name's Rick Moss, and I'm the National Director for Strategic Partnerships at Defeat Duchenne Canada. But before we get started, we've got a few housekeeping items that we want to go through. We remind you to please use the chat feature to post comments and questions. Our speakers will aim to answer them live at the end of the session. We also have some questions from families who were not able to attend today that we'll be dealing with at the end of the session as well. And speaking of the end of the session, we'll have plenty of time for discussion. Raise your hand to unmute and turn on your camera for in-person conversations with today's speakers, or post questions into the chat to be read aloud by myself or the moderator. We want to ensure you can clearly hear today's session, so if you would like to test your audio, please click on the up arrow next to the audio button on the bottom left-hand side of your screen, and this will allow you to adjust the volume level. For those of you who are new to our organization, it was founded in 1995 by John and Jesse Davidson, and known worldwide as Jesse's Journey, until we rebranded in 2022 with Defeat Duchenne Canada, a new name with the same vision, a future without Duchenne. Through our mission, we're focused on education and support for families living with Duchenne and Becker, advocating for access to treatments here in Canada, and funding more than $17 million to research projects around the globe. As mentioned earlier, today's webinar is one in a series of our educational activities across the year to help educate and inform Duchenne and Becker families about activity here in Canada. And none of this would be possible without the support of some of the corporate sponsors that you see listed here as our 2023 education sponsors, including Dyne. Thanks to PTC as our platinum sponsor, Edgewise as a gold sponsor, and silver sponsors, Regenex Bio, Atel Pharmaco, PepGen, Pfizer, Avidity Biosciences, and Roche. And now it's time to introduce today's session. We're pleased to welcome from Dyne Therapeutics, Dr. Ash Dugar, who will be more formally introduced in a moment, and introduce Molly White, who has extensive experience in patient advocacy and community engagement to her role at Dyne, where she leads the global patient advocacy and engagement across all the company's disease focuses. She previously served as Chief Executive Officer of the Myotonic Dystrophy Foundation and has been instrumental in advancing Dyne's clinical trials in Canada, including right here in London, Ontario. Presenting today on behalf of Dyne is Dr. Ash Dugar, who brings to Dyne over 20 years of broad experience across medical affairs, clinical and commercial development, and real-world evidence generation. Prior to joining Dyne, Dr. Dugar served as Vice President and Global Head of Medical Affairs at Sarepta Therapeutics, Inc. Previously, he spent 13 years at Pfizer, Inc. in various roles, most recently serving as Vice President and Global Head of Clinical Development of Medical Affairs of the Biosimilars Business Unit. We are delighted to have you here today to make this presentation, Dr. Dugar, and now I will turn things over to you.
Wonderful, thanks, Rick. So hi, if we can switch over to the title slide. Wonderful, thanks, Tiffany. So as Rick said, thank you for the kind introduction. I'm Ash Dugar, the Chief Medical Affairs Officer at Dyne Therapeutics, and joining me is Molly White, our Global Head of Patient Advocacy. First of all, I'd like to thank Rick and the organizers of Defeat Duchenne for inviting us to present at this webinar to update the Duchenne community on the progress of our program, and specifically Dyne 251, our clinical candidate designed to treat patients with Duchenne who are amenable to exon 51 skipping. As always, it's a true privilege to speak with the Duchenne community, and certainly the history of this organization is extraordinary in what they've done to engage with the community and advance research. Next slide, please. These are our forward-looking statements. Please refer to our website if you're interested in more details. Next slide, please. I always like to start with this slide. At Dyne, it all begins with our mission. We are laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is core to who we are and what we do every single day. And today I'm incredibly excited to inform you that we've been dosing participants in our Dyne 251 program. And our focus today is on sharing an update on the clinical trial progress and more details about our trial with Dyne 251. Next slide, please. In just a few years, we've built a robust portfolio focused on rare muscle diseases. Myotonic dystrophy type 1 and FSHD represent opportunities to develop first-in-class therapies. And in Duchenne, we're building a large portfolio starting with our exon 51 program. And we will follow this with programs for exons 53, 45, and 44. Next slide, please. So we're building Dyne as the leading muscle disease company to deliver for families living with DMD and other muscle diseases. Shown here are our 3 core strengths to help us deliver on that goal. First is our commitment to delivering for patients. We've initiated our clinical development program in Duchenne, which has been informed by the patient community and scientific leaders in the field. Second, we have exceptional in-house and advisory expertise with deep knowledge of DMD and developing novel therapeutics. We also have a world-class scientific advisory board, which includes luminaries such as Professor Francesco Montoni and Dr. Lou Kunkel. Third, we intentionally designed our program to overcome one of the biggest limitations of current treatments, which is to effectively and efficiently deliver modern oligonucleotides to muscle tissue. Dyne 251, our investigational medicine, is designed to utilize an exon-skipping phosphorodiamidate morpholino oligomer, or PMO, to increase dystrophin expression with the goal of significantly altering the course of Duchenne. We believe our technology can better deliver to muscles, including skeletal, respiratory, and heart muscles. And our technology should allow for redosable, titratable therapy with relatively infrequent dosing. Next slide, please. So I know that I don't need to spend much time at all describing Duchenne to this audience. We recognize that DMD is a degenerative condition where the lack of the dystrophin protein leads to muscle wasting. And eventually those muscle fibers are replaced by fatty or fibrous tissue over time. And while symptoms are seen as early as age 2, on average, Duchenne isn't diagnosed until age 4 or 5, and most individuals with DMD are non-ambulant by their 20s. We have learned so much more from you, the community, and advocacy leaders about how this affects families physically and emotionally. And it's you and your loved ones that have really taught us so much about this journey. Next slide, please. So this slide and the next few depict the role of exons in producing the dystrophin protein and how exon skipping works. So here you can see a length or section of DNA is called a gene. And each gene is made up of smaller parts called introns and exons. The introns are not shown on this slide. Now, when the body needs to make a protein, instructions in the DNA are given to a smaller molecule called RNA. And during this process, the introns are removed and all the exons are linked together to make one long chain of instructions, which are then carried to another part of the cell by something called messenger RNA. Once there, particles in the cells called ribosomes read the instructions, and this is a reading frame shown here to make the dystrophin protein. The mRNA illustrated here comes from the DMD gene and contains 79 exons that are linked together to form the instructions for making dystrophin. Now, mutations or errors in the gene alter those instructions for making dystrophin. And so types of mutations can include deletions of an exon or multiple exons, duplications of exons, and other changes. The most common mutation is a deletion of one or more exons, and this leads to the body not being able to produce the dystrophin protein. Next slide, please. So on this slide, we better illustrate, we better show this by focusing on a type of deletion mutation. You can see that we show a section of the 79 exons, exons 50 to 57. And if an individual exon is missing, for example, exon 52, the exons no longer fit together, and this is called an outer frame mutation. And since the pieces no longer fit together, the machinery that reads the exons to allow dystrophin to be produced cannot read through the chain of exons, and this results in Duchenne. Next slide, please. So the goal of exon skipping is to allow the body to make the dystrophin protein by skipping an exon to allow the remaining exons to fit together. Exon skipping splices or cuts a gene and restores the reading frame that I referenced earlier, acting as a kind of molecular patch, if you will. So exon skipping encourages the cellular machinery to skip over an exon by masking it with an antisense oligonucleotide or an ASO, which are small pieces of synthesized DNA. In this example, you can see a type of ASO, as I mentioned before, called a phosphorodiamidate morpholino oligomer, or a PMO, that can skip exon 51 to allow the remaining exons to fit together. This allows the production of a shortened functional form of dystrophin that the body can produce. Next slide, please. So to date, one of the biggest challenges to developing highly effective exon skipping treatments has been the lack of getting enough of the PMO into muscle cells, such as skeletal muscle, diaphragm and respiratory muscles, and heart muscles. And Dyne is looking to overcome that barrier, and our scientific platform is expressly designed to address this issue. Next slide, please. So here I'll take a few minutes to describe the FORCE platform. The FORCE platform, as I mentioned, drives our efforts to develop and deliver muscle-targeted therapeutics. As I mentioned, it's expressly designed to address one major issue in muscle diseases, which is efficiently and reliably delivering ASOs that target the genetic basis of diseases to those key muscles, like skeletal muscle, cardiac, and smooth muscle. As you can see, Dyne's FORCE platform consists of three parts: a proprietary antigen binding fragment, or what we call a Fab, to enable targeted delivery of the ASO to muscle, a clinically validated linker, and an ASO selected to target the genetic basis of disease. Now, I'll start by describing these three components, and then I'll come back to the importance of targeting the transferrin receptor 1, or TFR1, to be able to drive targeted delivery of our investigational medicine to muscle. On this slide, you can see a Y-shaped structure, which is a monoclonal antibody. So we chose a portion of this, our Fab, as I mentioned, because we believe it offers powerful advantages over the larger monoclonal antibodies, including its smaller size for better muscle penetration, lower overall protein load, and the potential for enhanced tolerability. The goal is ultimately to enable targeted, effective delivery of therapeutic molecules to skeletal, cardiac, and smooth muscle. We selected the Val-Cit linker, shown here at the bottom of the slide, for our platform based on stability and its clinically validated safety and efficacy in approved products. So this linker is used in products that are currently on the market. We believe this further de-risks our platform. Now, we're utilizing the same Fab and linker across all our programs. The versatility and modularity of our scientific platform provides flexibility to deploy different types of antisense oligos, different types of ASOs that are rationally selected to match the target biology for each disease. Where dystrophin restoration is the key, as is the case for Duchenne, it's critical that we select an exon-skipping PMO that targets the specific mutation in the DMD gene. Now, the mechanism of FORCE is different from other approaches. We're essentially using the cell's natural biology to deliver our medicine to muscle cells. You heard me say before that our Fab binds tightly to something called the TFR1, or the transferrin receptor 1. Well, TFR1 receptors are highly expressed across the surface of muscle cells, including skeletal muscle, respiratory muscles, cardiac, and smooth muscles. So the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our ASO to enter those muscles to target the genetic basis of disease at the level of the nucleus. So we're able to harness what already exists on the surface of muscle cells to deliver our therapeutic agent to muscle with this receptor-mediated mechanism. By targeting muscle specifically by using TFR1, we have the potential for a wide safety margin, a wide therapeutic index, resulting in less frequent dosing and chronic administration. Next slide, please. So in this next section, I'll talk about some of the work we undertook prior to entering the clinic with Dyne 251 that gave us confidence to move into the clinic. Next slide, please. So at Dyne, we've not shied away from using multiple preclinical models available to us to characterize the FORCE platform and gain a deep understanding of our platform and Dyne 251, our investigational medicine. This work has been critical to inform our approach into the clinic and ensure that we have patient safety first and foremost in mind as we put together and executed our clinical trial. We've looked at non-human primates or monkeys to help us understand how much Dyne 251 gets into their bodies and how much exon skipping occurs. Because NHPs are not a Duchenne model, we can't look at dystrophin production in this model. We've also looked at a standard mouse model for DMD, the MDX mouse, which has a mutation in exon 23. And we've also looked at patient cells. I'm going to share just a little bit of this data with you now. Next slide, please. So on this slide, we show our FORCE platform in the MDX mouse model, again, a standard animal model in Duchenne. These MDX mice have a mutation in exon 23. You can see FORCE resulted in robust and also durable dystrophin production across key muscles affected by Duchenne. So in this study, with a single dose of 30 milligrams per kilogram, we evaluated the animals 4 weeks later. The top half of this slide shows some of our data demonstrating robust levels of dystrophin expression across key muscles. We were able to get to very high levels of dystrophin expression, about 50% in the quadriceps, 90% in the diaphragm, and about 80% in the heart. We are especially excited to see these levels of dystrophin restoration, especially in the heart and diaphragm, where current PMOs have had significant limitations. On the bottom half of the slide, we show dystrophin localization in the sarcolemma muscle membrane after just that single dose. Now, this localization is important to be able to give us confidence in potentially seeing functional benefit. We saw profound results, including about 70% dystrophin positive fibers in the quadriceps and about 80% in the diaphragm and the heart. Next slide, please. On this slide, I'm pleased to show newer data that was presented at the MDA Clinical Conference in March. Now, the D2 MDX mouse is a relatively newer preclinical model consisting of the skeletal muscle pathology associated with Duchenne. Specifically, these mice have characteristics of a severe disease phenotype. These animals exhibit significant muscle damage, impaired muscle regeneration, and significant fibrosis, among other features. And it's a very relevant animal model in DMD. So here we show data using the FORCE M23D repeat monthly dosing. And we're looking at, on the left-hand side, dystrophin localization at the sarcolemma. And on the right-hand side, we're looking at progression of fibrosis in these D2 MDX mice. So, just a little bit more on the study design. We looked at the D2 MDX mice with a vehicle or essentially a placebo and repeat monthly doses of 30 milligrams given earlier in the life of the mice and a bit later in the life of the mice. And we evaluated, as I said, dystrophin localization to the sarcolemma and then progression of fibrosis. So on the left-hand side of the slide, we compare these wild type or healthy animals to the D2 MDX mice treated with vehicle or dosed early or late with FORCE. And what we see is robust localization of dystrophin to the sarcolemma which, as I mentioned before, gives us confidence that there should be functional benefit. On the right-hand side of the slide, you can see at baseline the level of fibrosis at 5 weeks and 22 weeks. And with both early and late force administration, you can see the change and the change of the progression of fibrosis. So we're actually changing the trajectory of the development of fibrotic tissue. Not surprisingly, earlier treatment is more beneficial than later treatment. Next slide, please. Next, we evaluated Dyne 251, our clinical candidate, in NHPs or in monkeys. Now, these studies are a key step in generating preclinical data before going into clinical trials. We measured exon skipping in the quadriceps, heart, and diaphragm. And just as a reminder, in NHPs, we cannot look at dystrophin restoration because these are healthy animals. We can look at exon skipping. The key message here is that with our FORCE platform, time on therapy matters. Over a 2-month period, we observed robust exon 51 skipping across muscles. After 5 weekly doses at 30 milligrams per kilogram, Dyne 251 demonstrated about 50% exon skipping in the diaphragm, about 40% in the heart, and about 20% in the quadriceps. We also evaluated Dyne 251 in 5 and 13-week toxicology studies in monkeys. In both studies, we saw that Dyne 251 had a favorable safety profile with no dose-limiting toxicities observed up to a maximally feasible dose, which means we dosed monkeys as high as we could formulate the drug, and we did not see, and it was safe and well tolerated. So given the safety profile, we had a lot of confidence as we moved into the clinic. Next slide, please. In this next part, I'll talk about our phase 1/2 study called DELIVER. Next slide, please. So prior to starting our Dyne 251 clinical development trial, we proactively obtained expert feedback. We engaged key opinion leaders or KOLs from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. And importantly, we gathered this feedback from across the globe. KOLs both in the US and outside of the US with experience running early and late stage clinical trials have provided advice on our overall clinical development program, including our clinical trial design, patient population, biomarker and functional endpoints, and key safety considerations. Very importantly, they have helped us extract learnings from the many natural history studies in this field. It's clear that KOLs believe dystrophin continues to be a key endpoint to assess for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders, such as those on the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries, including Canada. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design and has included, but it's not limited to, key considerations for choosing a clinical trial, review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. Based on this extensive feedback, we developed our clinical trial for Dyne 251 in patients amenable to exon 51 skipping. And I'll now just describe some of the key elements. Next slide, please. Our clinical trial for Dyne 251 is a global trial consisting of essentially 4 periods. We have a 6-week screening period, followed by a 24-week placebo-controlled multiple ascending dose period where individuals receive either placebo or Dyne 251 administered every 4 weeks. This is followed by a 24-week open label extension period where all participants will be escalated to the highest safe dose of Dyne 251 administered every 4 weeks, and then finally to a 96-week long-term extension period. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We plan to evaluate the following outcomes in the DELIVER trial: safety and tolerability, of course, dystrophin by Western blot, muscle function relevant to both ambulatory and non-ambulatory participants, using outcomes such as the North Star Ambulatory Assessment or NSAA, and performance of upper limb function or the PUL 2.0 to help us understand changes in lower and upper limb function. We will also carefully assess lung and heart function. And importantly, we will be dosing monthly in the clinic and have the potential to explore even less frequent dosing than that. With the help of our expert advisors, we're very confident that our study design is robust, will measure key outcomes, and is striving to manage the burden on families. I'm also pleased to say that we do intend to communicate initial data with respect to safety, tolerability, and dystrophin in the second half of this year. Next slide, please. This slide shows a select set of inclusion and exclusion criteria. Much more detail can be found on our clinicaltrials.gov page, including open clinical trial sites. We're enrolling patients with Duchenne who have mutations amenable to exon 51 skipping therapy. Our goal is to enroll approximately 48 male participants ages 4 to 16 years who are either ambulant or non-ambulant. We do require open biopsies of the upper arm muscle, and participants should be receiving stable glucocorticoid dose for at least 3 months before the start of study drug administration. And if someone has been on exon skipping therapies, we do allow those patients in our trial with a 12-week washout. Importantly, this is a global study. We are planning to have sites in Canada. We have a site currently recruiting in Canada with Dr. Craig Campbell at London Health Sciences Center. And we also plan to open up a couple of other sites in Canada. Again, you can go to the ct.gov website for more details on inclusion-exclusion criteria, as well as a listing of currently recruiting sites in Canada, the US, and around the world. Next slide, please. And finally, we've tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We are working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and the sacrifices that you continue to make. On behalf of Dyne, I would like to thank you all for your time and attention in listening to this presentation. And Molly and I are pleased to take any questions.
Dr. Dugar, thank you so much for the update. And that was a very informative presentation for our families. I think the results are very encouraging at this stage, and we're very anxious. Of course, time is always the big factor to find out where things are headed. I mentioned just before we started the webinar this afternoon that we'd had some families be in touch regarding questions, and I know you have answered some of the questions I'm about to ask, at least to a point, but I want to ask them on their behalf. This is a 3 or 4 part question. The first one I have here for you. How long does the treatment last? Would this have to be repeated? Which I know some of the questions, as I say, you've already dealt with, but is it a single treatment only, and does it preclude you from additional treatments?
Yeah, great questions. And I'll try to provide some more clarity around this and then invite Molly to also chime in. So this is a chronic treatment. So it's an exon skipping modality. It is, we have developed this purposefully for chronic administration. It is an IV infusion administered every 4 weeks. And again, we are exploring even less frequent dosing than that. We do believe that this has several advantages over other modalities. If I think about maybe where your question is going, it is certainly not a one and done therapy. It is for chronic treatment, and the good thing is it is titratable. It's tunable. It is something that we understand the onset within the body, the onset kinetics, and we're gathering more data as we get more data from the clinic. And then also the offset. And all of this information from the preclinical work to the clinical data that we'll be generating helps us continue to refine our dose and dose regimen. And that is really one big goal from this particular study. So, we think there's a lot of advantages to being able to have something that you can, frankly, have in your system. And if you need to, sort of go to some other frequency, you can go to another frequency. So it's not, again, it's titratable. So it's very different than gene therapy, which is maybe where the question is also coming from. Gene therapy is a micro or mini dystrophin. It is you can't fit the whole dystrophin gene in the viral capsid. And so, each company that is developing gene therapies has had to make choices about what they package into this virus that is then administered. And at least currently where technology exists, redosing is not possible after receiving gene therapy. And so there's a lot of limitations in terms of pre-existing antibodies, right? The immunogenicity related to the viral capsid and in some patients to the transgene. It's not something where it can be redosed, though there is work going on to see how one can get redosed for gene therapy. So very different, from that kind of treatment. Molly, do you want to add anything?
That was pretty comprehensive.
Yeah. No, I think that does answer all the questions. There was a 3 or 4 part question there, but the other question is, my son's deletion is not 51. I'm wondering why so many treatments are just for the exon 51 deletion. That's part 1 of it. And I know you've mentioned there are other deletions you're going to be working on in the presentation, but they didn't have the benefit of hearing that. So they're asking, why does it always seem to be exon 51, I guess? Yeah. And what would or could this mean for other deletions?
Yeah, absolutely. Great question. I think that is probably the number 1 question that we get. So I'm going to answer it in a few different ways. So first of all, from an epidemiology point of view, that exon 51 population is about 13% of the DMD group. And so from the point of view in being able to run a clinical trial, there are more patients available to be able to run a clinical trial, in terms of having patients who have exon 51 amenable mutations. That's part of it. The other part of it is that let's remember, aside from the US, there's only maybe there's only a couple of other countries where there is an exon 51 therapy approved. Unfortunately in Canada, there is no therapy available at all for Duchenne except for steroids, and maybe a few other things for symptomatic treatment. But really, there's no disease modifying options here, and so we can apply that sort of globally. There is a huge unmet need within the Duchenne space. The exon 51 population is sort of the biggest in that space. We can run those clinical trials and try to advance a truly potentially transformative treatment. Right now, the bar is 0.3% dystrophin. With current, the current PMOs that are amenable to exon 51 skipping that are, approved in the US, and that's over a period of 6 months. We just need, which is fantastic. And it's good that there's something out there, but we need better treatments. So the other interesting thing about the question is there's a bar, right? There's a sort of a bar that has been set. And I think it's important that companies are able to develop treatments that far exceed that bar to give confidence in their scientific platform, in their approach for being able to solve this disease. And so you do see a lot of companies going after exon 51 because there's a bar that's been set. And so if you can sort of really leap over that bar, your scientific platform is meaningful, and then you could sort of run into other exon skipping drugs, just like we're planning to do.
Great, thank you. A couple of, and again, remind anybody who's on, if there's any questions, please, pop them in the chat or put yourself on, and we'll get to those. A couple of things I observed in the presentation, and this may or may not be a fair question, but some of the results were terrific. I mean, the results are terrific, and the response has been terrific in the models that have been used, the monkey and the mouse. How does this compare industry wide? Is that a fair question? I mean, the results really do seem impressive from what's happening as far as dystrophin is concerned.
Yeah. So I think that, the way that I'd answer that is, we are really pleased with the level of exon skipping in different animal models or different, non-clinical models. And where relevant, the level of dystrophin restoration and dystrophin positive fibers and this sort of change in trajectory of fibrosis in muscle tissue. So we believe that we have a pretty unique non-clinical data set that sets a bar in the field in terms of the non-clinical data for a given platform from companies. Now, what we want to be careful of is doing cross-study comparisons. And so, unless there are studies where you have exactly the same molecules in the same animal models, evaluating the same muscles at the same time points, and you run those studies, it's a little fraught in terms of doing a comparison across preclinical data sets from different companies. So, what I'd say is, when we look at our data, we believe we stand out. We're able to get into muscles where other modalities have not been able to get into, like cardiac muscle,
Yes.
and the diaphragm as well, where there hasn't been great penetration from other modalities. I think the other differentiating feature is the receptor-mediated delivery. So using the transferrin receptor and our Fab, the piece of the antibody, and its clinically validated linker, we're able to really utilize the body's natural cell biology to be able to drive the ASO into the nucleus, unlike other technologies where there needs to be membrane destabilization, where there are some of the other technologies, cell penetrating peptides and other technologies that work differently, that are in the clinic, that have had issues in terms of off-target effects. So again, we think this active receptor-mediated delivery has been at least proven in our preclinical models to do what we thought it would do, and hopefully now we can replicate this in the clinic. And I think that's the biggest thing: can we replicate what we've seen in the animals in the clinic?
Great, thank you very much for that update. This may be more of a question for Molly, but the trials and the Canadian trial sites and so on and so forth. Can you give us just a little more insight as to the timelines and parameters, where you're at and where you want to see those go?
In terms of the scheduling of when sites are opening, and
Yes.
I'm going to pivot that back to Ash.
Are you okay?
Because there's only so much we can actually share.
Okay.
Yeah. So we do have. So first of all, just having been in the space for a while, I think it was important for Molly, myself, and Dyne to be able to serve the Canadian community.
I appreciate that.
I think the Canadian community has been underserved in the space for a long time.
We would agree.
Yeah.
Thank you.
So Dr. Campbell, who is also a great advisor and has helped us tremendously, is a currently active site. We have 2 more sites in Canada that we are working with to open up as soon as possible. And that is the current plan for this phase 1/2 safety tolerability study. As you would imagine, it's not a lot of patients. You don't need a ton of sites. And so our plan is 3 sites in Canada. But of course, assuming things go well, we will then have larger trials that we're going to have to open up as well, in terms of randomized in terms of larger placebo-controlled randomized studies. And we'll look to potentially expand in Canada from there. So what I would say is stay tuned maybe over the next, 2.5 months, 2 to 2.5 months to see hopefully a couple more sites in Canada posted up on clinicaltrials.gov.
Perfect. And of course, we have our Canadian clinical trial tracker where we'll be putting that up as well. And as you know, anything we can do to help support that, we're happy to lend any support and be in touch with our Duchenne Canadian community, whatever it takes to make it happen. And then perhaps the last final question here may be on the initial data release at the second half. We've got, just let me just jump to one question we've got in the chat.
It's a great question.
Yeah.
Will boys who are taking another study drug like vamorolone be allowed into our study? Yeah, it's a really important point. So as you would imagine, as I've said, this is a safety tolerability study. And so when we do studies like this, it's important to try and make sure that we understand what the benefits and risks of Dyne 251 specifically are. So our inclusion exclusion criteria currently requires a washout of other study drugs. So for someone who is on vamorolone to enter our trial, they would need to wash out of vamorolone because it is not an approved, standard of care, at least at this point in time, not an approved and standard of care steroid, right? Like prednisone or deflazacort. So, but again, as vamorolone gets established as standard of care, then we'll have to reevaluate.
Great. And where I was going was you mentioned initial data the last half of this year. The source for that will be
the source. Can you hear me?
The data will be coming from.
Oh,
I mean, what data?
Yeah. So it'll be coming from Dyne. Yeah. So we will be communicating initial data. We haven't specified anything further than the second half of 2023. It'll be safety, tolerability, and dystrophin. It'll be coming from us. And what Molly, I think maybe where you're going, what Molly always reminds us is how we communicate with the community is absolutely critical, right? That the community should be hearing, about the data from Dyne, not through, some other means. And so we are committed to doing that. Molly and I, we're and the company, we're committed to making sure that, we are the primary communicators with respect to data to the community. And hopefully we have a decent track record of that as we've gotten non-clinical data out there. We try to get in front of the community within, 24 hours,
Right.
Molly sends announcements out. I try to do webinars. Molly and I try to get out there to the community. So you're gonna see, more of that, from Dyne and from us.
That's terrific. And one of the things we keep hearing, and I'm sure from every community, whether it's the US, Europe, Canada, wherever, is just a lay explanation. Of course, easier said than done, I know, but for the scientific information is critical, of course, and the comparatives and all those things. But also, I think that one of the key things our community is looking for is, what does this really mean? How does this potentially impact my son? That's the basic question that we start from. And how will this impact my son? What's the potential that it has to help the treatment? So those things you guys have done a great job of in the past, and the communication's been great. We want to get the information out as quickly as possible, of course, and by having something like that, it accelerates our ability to share that information quickly on socials and on our website and so on and so forth. So thank you.
Yeah, of course.
We're putting a whole plan together for this. So we'll be in touch with you so that as soon as we are able to announce something. I mean, we have typically gotten, as Ash said, communication to the community out within 24 hours of releasing it publicly at all. So, I'll work with you ahead of time. We'll figure out the best strategies for getting it out. And
terrific. Well, you folks have been a pleasure to work with,
and
this has been great. Thanks everybody. We have a few announcements we're going to make before we wrap things up, but I do want to thank the team on screen here. Molly, thanks so much. And Ash, terrific. Behind the scenes, Kate and Tiffany, great job. I do want to just share some closing comments though. So thank you to everyone for participating this afternoon.
Thanks,
Thank
Rick.
you. Thanks for
Yeah,
inviting
thank
us.
you. Our monthly community social is tonight, as a matter of fact. Canadians Talk Duchenne. It's a social for parents, caregivers, and those living with Duchenne. And tonight we'll hear the struggles and triumphs of living with Duchenne from Anne and Riley in British Columbia. Our community annual general meeting is set for Monday, June 5th. Reminder, there's still time to register to join in person or virtually, and you can do that on our website. If you have not connected with us in the past, the emergency care resources are available, and we have them up on the screen for you. They're free and available to anyone in your family who may be concerned when caring for anyone with Duchenne. I also want to remind everybody that we have a clinical trial finder. We mentioned it just a few minutes ago on our website, and this is becoming more and more important as different pharmaceutical companies find us, and we work with them in trying to set up trial sites. And it has a Canadian spin on it, which is critical, of course. It's available on our website or by going to defeatduchenne.ca/clinical finder tool. And a reminder to stay in touch with us through your social media channels. And don't forget to subscribe to our e-news to hear the latest on research, advocacy, and education. And speaking of education, I want to get a plug in for our family forum, which is set for November 4th and 5th in Ottawa. It's an annual event. It's an educational event. Registrations will open on our website next week. It's going to be held Saturday, November 4th and Sunday, November 5th at the Marriott Hotel in Ottawa. So stay tuned to our website and your socials for updates on that. Thank you to everyone, especially the Dyne team, for a great presentation today. Lots of information exchanged. This has been recorded. Share with family and friends at a later date. We also want to thank everyone who joined us across the community today. Thank you so much and enjoy the rest of your day.